Well, thanks, Rennie, for inviting me to this. My name is Simon Eickhoff. I'm an MD by background and did my doctoral work in neuroanatomy with Carl Zillis. So I started out very classically trained, then moved into imaging with mainly functional MRI, then became more interested in clinical applications and actually spent 3 years as a psychiatry resident from 2009 to 2011. So at that time, I've moved away from neuroanatomy more into neuroimaging and psychiatric applications. And then after a few years, I dropped out of the clinical part and focused fully on research. And also the work changed with now computational methods, connectivity, and in general, machine learning becoming a bigger part. And that for a long time was mainly on unsupervised methods. So for many years, I've done a lot on clustering and factorization. And it's really over the last, I would say, roughly 5 years that I got more into what's more commonly known as machine learning, namely supervised methods for prediction and classification. So where does that lead me? I do have a very broad background that's mainly a mix of medicine, neuroimaging, and machine learning nowadays. And I'm pursuing that at 2 different institutions, but it's actually the same group. It's the Institute of Systems Neuroscience at the Heinrich Heine University in Düsseldorf and the Institute of Neuroscience and Medicine at the Research Center in Jülich. So one is a classical university. That other, Jülich, is a non-university research center. And I guess for Americans, the best way to conceptualize Helmholtz centers is probably similar to the NIH intramural. So there is more core funding and it's more geared towards services and platforms rather than purely research output. And together that puts me into a quite interesting position that I'm kind of bridging the infrastructural aspect and the more psychology medicine aspect. Any questions? If not, I will actually fill that with life by giving you a few minutes of a presentation on quantitative markers of brain health. Where do we want to go? So one of the key things that everybody who's ever worked with neuroimaging data will immediately acknowledge is that there is a massive inter-individual variability. No matter what you look at, structure, function, connectivity, no matter what your parameters, what your indices, they all vary considerably across individuals. And hence, I think one of the key challenges for the field is to find out how this inter-individual variability relates on one hand to factors such as age, gender, genetics, but critically also experience, health, and lifestyle, which we would all suppose to shape brain variability. And then on the other hand, how does it relate to what we could call the downstream effects? Cognitive performance, socioaffective functions, psychopathology, or very broadly, behavior. Why is that interesting? Well, it's first of all, obviously, there's an academic interest because we would like to know how the brain is organized, how it relates to behavior, but there's also, and I think particularly also in the current context, there are very intriguing perspectives then in the application level. The overarching long-term idea is that somebody comes into a hospital or into a checkup center, has a battery of imaging assessments, and from that, we could, for example, then forecast what kind of a therapy would be most suitable for that particular patient. Or we could actually have an idea about what is going on in the future. This is critical both in clinical medicine, but also in the context of preventive procedures, because the key idea here is that there may be neurobiological change, damage, that is yet very well compensated. So that's one of the fascinating aspects about the brain. It can compensate structural functional damage surprisingly well until the point where the reserve capacity is basically exhausted. So could we, for example, make statements about the expected progression in motor decline? And for example, in a simple traffic light system, say, well, there's a moderate risk. What's the risk for dementia? That's very high. What's the likelihood of mood disturbances? We don't see anything in the brain scans that allows us to predict a higher risk. Now, these are kind of like long-term perspectives. What can we already do quite well nowadays? We can first of all assign, diagnose a new individual with a, I would say, decent confidence as either patient or control. So we make diagnostic classifications. And I think that's one of the most fascinating topics. We can also predict phenotypes on the individual level. And one of the interesting things that we see here is that usually there is a divergence. So we would, for example, say in this particular person, the brain says this should be the phenotypical expression, the predicted one. And then we can measure it also using standard neuropsychological or clinical tests. And what I'm particularly intrigued by is potential discrepancies here, because they would indicate that even though, for example, cognitive function is still normal, in the brain, it may look worse. Give you a very simple example. You have two patients, two people that come into the hospital and worry about cognitive decline. And for both, you predict cognitive functioning based on the brain scan. And both patients have the same phenotypically measured level. They are both at the same level cognitively. But in one person, the brain scan says, you should be much better. In the other one, it says, you should be much worse. And that could be an indicator that the person that was doing better than the brain indicated is compensating very well, but maybe at a high danger of deteriorating, whereas somebody who should have done better based on the brain scan maybe just had a bad day, but still has a lot of reserve. So this is the long-term picture, the overarching goal of what we, and I guess some of you and many other people in the field are looking at. Let's go to some concrete examples. And I roughly structured this talk into two domains, structural imaging and functional imaging. And now we start with the structural images, which I am actually a big fan of because structural images are very standardized, very reliable, and very accessible also on the clinical side. So here you see two brain scans. One on the left is from a young, healthy adult. And now you look at please at the scan on the right, and you see there's quite a few differences. The sulci are much wider. You can see the ventricles much better than on the left. And also, if you look more closely, you see that the cortex seems to be quite thinned out. This is normal aging. On the left, the subject is, I think it was about 25. On the right, you have mid-70 person. So what you see here is actually the basis of what you read in the radiology reports. A trained radiologist can tell quite well whether a scan looks age equivalent or pre-age. Now, what happens when we look at it with classical statistics? Now, here we correlated regional volume, area volume by age. You see there are quite strong associations. And in fact, if you go on the strongest one, you have a significant correlation of R equals minus 0.6 between chronological age and local gray matter volume. This is highly significant, not very surprising, but it also outlines the key problem that we have with classical statistics. If you have somebody of a certain age, there's a big range in brain volumes, and vice versa, a certain volume may belong to people of a very broad age range. And in fact, even a highly significant correlation at minus 0.6 only explains roughly 40% of the variance, which is very nice if you do population statistics, but doesn't help you at all when you're actually trying to learn something about an individual subject. So we need to change our perspectives here, change away from classical inference towards machine learning. Now, what is the key idea? I will show you the overall setup in a minute, but I want everybody to take home what is the one key difference. And the key difference is that we are not interested in how well our model fits the data that we have, but how well does the model capture new data? So the key difference between statistics and machine learning is not about the algorithms. A linear regression is a perfectly fine machine learning tool. The key difference is the distinction between within sample statistics, how well does our model fit the data it was trained on, fitted on, versus out of sample validation, how well does the model capture new individual cases. And from that, let's say, very placative distinction, it should be quite clear that most of the applications that we and others, we all are interested, are not attainable by classical statistics. Biomarkers that are actually put to use on the individual needs to be evaluated on the individual. So what's the overall pipeline? We have a lot of imaging data that is then pre-processed. That in itself is, I think, a topic that deserves almost its own session, its own workshop. Data management, data curation, automated pipelines, and quality control. It's actually probably the most critical part of all of our endeavors, in particular when we are looking at large samples of thousands, if not tens, hundreds of thousands of subjects. Then we need to acknowledge that even if we have large samples, we are still having a rather poor feature to subject ratio. Why is that? If you look at the right brain here in the black box, that's a standard T1 voxel-based morphometry. These are actually 200,000 individual data points. We look at it as one image, but it's actually 200,000 features. So even if we have large samples, let's say 50,000 subjects, we are still in a poor feature to sample regime. Key point here is, though, we don't need to be, because although we have 200,000 different data points, we don't have 200,000 information. These individual voxels don't covary independently by itself, but rather we know that the brain is organized into distinct modules, brain areas, and so we can actually represent this very high dimensional information in a much more compressed format. When you see that on the lower right, where we have for each subject then only the volume information for several hundred areas. And this is a critical step. We get away from, let's say, 5,000 subjects by 200,000 data points and move into a 5,000 subjects times 600 data points regime. So we come from a horrendous feature to sample ratio into something which is actually recommended and stable. So the rule of thumb is you should have about 10 times as much observations than you have features. Now we can train a model, finding the relationships between the matrix on the lower right and whatever target phenotypical variable we assume. And once we've trained that model, we can then evaluate it on new subjects to see whether we can indeed predict the phenotype in that individual new subject. Any questions on that point? I will walk through a few examples in the next slides, but for me, it's most critical that everybody is on board with this general outline, with this general idea, the importance of out-of-sample validation and the critical aspect of feature to sample ratio. That's really, if all of you only, in particular those that come from a different background, only remember these two things, the talk was successful. Okay, let's go into the standard application, I would say. And it's not only the standard, but I think also very relevant in our current context, which is aging assessment. Can we predict individuals' age from brain scans? Now, why has that been the go-to model that a lot of the algorithms are tested on? Well, there's one very practical reason. Age is something you can really get in very large numbers. You can get age plus T1 scans from thousands of subjects. So we just ran it on a few, I think it was about 5,000 subjects. What you see here now on the x-axis is the actual chronological age of the subjects. On the y-axis, the age that was predicted by the model from the T1 scans. And as you can see, we can actually estimate individual age fairly well. On average, the precision is about 4 to 5 years. Now, if you look through the entire literature, you will see that there is some variability in the performance estimates you get, but it seems that roughly 4 years, give or take, for lifespan or adult lifespan data is, give or take, what most algorithms can do. You can do better if you have a narrower age range. So if you, for example, only deal with kids or only deal with elderly subjects. And it has been suggested by a preprint from the UK Biobank that if you go into a much larger training data set, like several 10,000s of subjects, you can shave off another year or two. But give or take, I think we can assume that in population multi-site samples, you usually get around 4 to 5 years precision. Now, the question is, does the difference between true chronological age and estimated age, does that mean something, or is it just random noise? One good test case here is to look at cases where we know that there is accelerated aging and brain atrophy. So here you see a sample of healthy controls. And what you see on the y-axis is the brain age deviation or brain age gap. A positive number means your brain looks older than you are. A negative number means your brain looks younger than you are. And as you can see for healthy subjects, that centers around 0. If we look at patients with mild cognitive impairment, though, we see that their brain, on average, looks about 5 to 6 years older than they really are. Last but not least, if we look at patients with Alzheimer's disease, we can actually see a quite substantial increase in apparent brain age to about 10 years acceleration. Now, we replicated that on a different sample. And in this presentation, I actually cut out a lot of technical slides. I'm still going to talk 1 minute about it, if I may, because one of the challenges that are there is side effects. So you can, if you do a cross-validation within one particular site, let's say at MGH, you have 5,000 scans, all from the same scanner. And if you fit your models on that data and test it on left out parts of the same data, you can get beautiful results. And you take the same model and test it on our data, acquired in newly. and frankly, it most likely will fail rather graciously. So in this paper that I can really recommend everybody to look at, we tried to find a way around that. And the idea here was stratified subsampling. So you have many different training cohorts, and all of your training cohorts are biased. Some have more older subjects, some have more younger, some more males, some more females, and so on. And they all have different scanners. So the trick here is to not force feed everybody into the same model, but rather only pick a stratified subset, the same number of subjects per age bracket, per gender, per site. And then your training data set of like 4,000 subjects shrinks down to a few hundred. But it's a few hundred that are completely unbiased, because the model, the training algorithm, will not have any biased information, more females in that group, older patients in that group. And then you do that many times. And so you fit a lot of not so good, but completely unbiased models. And if you then pool them, you actually get a good, but still unbiased model. And that's what we did here. And what we then saw in that new sample is that, again, healthy controls have a BrainAge gap around 0. And that was the focus of that work here. We also looked at sleep disordered breathing. That's mainly sleep apnea, which is a risk factor for cognitive decline and dementia. Somewhat surprisingly, we did not find an increased BrainAge in people with sleep disordered breathing. We also nicely replicated the results for MCI with the new algorithm. Again, no effect of sleep disordered breathing. And also for Alzheimer's disease, nice replication, but no effect of SDB. So what I really like about that study is that it introduces a new methodological concept, then replicates a previous finding with the new method, and looks at a new topic. And I think we need a lot more of that in the literature, because 90% of the papers conflate a new method with a new finding. And that makes the literature extremely disjoint, but also fragile. Another condition that we looked at using the same approach is Parkinson's. We had 2 large samples. One was from the PPMI database, one is a local cohort, and a big data set of controls. So here in that slide, you actually can see quite well what I mentioned about bias. So these were all our training data sets. And you see, for example, the blue data set is very much biased towards subjects that are like in their 60s and 70s. Now, the problem is side effects are big. So it's easier for the algorithm to learn side effects than to learn actual age effects. We can avoid that though by the stratified method, and we can see that our healthy controls from the PPMI and from the local Parkinson's database, we could predict quite well and unbiased, so with a mean average error of about 4.5 years. And then we also saw some interesting effects in Parkinson's. What was interesting about that? Well, 2 things. First off, we do see an advancement in BrainAge in Parkinson's patients, but this is relatively minor. So on the local sample, we had about 3.5 years. On the PPMI, it was just about 2.5 years advancement. This is a striking difference to, for example, dementia, but also to virtually any other disorder. That's an interesting paper. There was a recent paper by the Norwegian group, Vesir and Kaufmann, that looked at a range of different disorders, suspiciously not Parkinson's, and found BrainAge basically being substantially increased in almost any disorder. It seems relatively minor in Parkinson's disease, interestingly. And we also saw that, somewhat surprisingly, the effects in PPMI were rather low. But this could then quite nicely be explained by the fact that PPMI is a progression cohort, so it's mainly very young subjects, early disease, and we saw that apparent brain age increase was related to both cognitive and motor scores across the cohorts. So we do have a marker of brain disease. It seems to also relate to brain function. And this is something we then tested in a population-based sample from nearly 1,000 brains. Again, our brain age prediction was about 4.5 years. Now, does it indicate cognitive score? So what you see on the right is the so-called Benton test. It's a very simple classical neuropsychological test that is a global marker of brain damage. And what we can see is that even if we adjust for basically everything, age, gender, IQ, and so on, we still find albeit rather weak relationship between brain age advancement and Benton performance. So if we sum all of that up, we can predict individual age rather well from T1 MRI. We now have a lot of tools and approaches that make it fairly robust against bias, including the dreaded sample or site bias. We see an increase in brain age in neurodegenerative disorders, but we also see a relation between individual brain age and cognitive functioning. Where does this lead us to? What's the perspective? One is, I think we need to get yet a better grip on the brain age gap. So which factors in life actually influence brain age gaps? What makes your brain younger or older? And this is obviously also a prelude, so to speak, to using other phenotypes, other behaviors as target. We are focusing on age because it's interesting, relevant, and we can do it well. But if we do actually have sufficient sample size, we can basically substitute any behavior of interest for age and use the very same modeling approaches. Coming to the second, somewhat shorter part, and that's about brain dynamics. So I am a very big fan of structural imaging because it's robust, it's easy, it's clinically doable, but it obviously ignores all the beauty that you see here, namely that there is a lot of coordinated dynamics going on while brains are either doing tasks or being addressed. There is a huge literature on task-evoked brain activation, about 30,000 papers. And if you look at the literature, one thing that again stands out, I think that is a recurrent topic on anything, is variability. So what you see here is the activation foci from about 200 fMRI experiments on working memory. And as I can hope most of you can appreciate, these are somewhere all over the place. But there are also some regions where they seem to be more frequent than in other places. So we can use meta-analytic models, here the so-called activation likelihood estimate, to build probabilistic models of brain locations. So where do working memory tasks activate the brain? And if we do then inference against the null distribution of random spatial association, we can actually get from this big and heterogeneous literature onto very discrete sets of regions. So there are hundreds, thousands of papers that report all sorts of tasks and contrasts, and we can consolidate that into rather discrete models of these are the brain regions that are most robustly activated when people do working memory tasks. The key idea then is to conceptualize these meta-analytic results as nodes of the relevant networks, and then we can go to individual data, individual resting state data, and extract your individual network-specific connectome. And this is, I think, a critical step, because if there's one thing we need to admit, task-based neuroimaging will not scale into clinical and cohort-based applications. But here now we have an approach to use very well-attainable resting state scans and still extract information on task-specific networks in these resting state scans. Now we can use that again for machine learning, and by doing so we can answer the question, does it actually matter where we look? So what you see here is a range of different meta-analytical networks, and we asked a very straightforward question. For each of these networks, we trained a machine learning model to classify Parkinson's patients from controls or schizophrenia patients from controls. Key thing here, it's the same resting state data sets. We are just looking at different parts of the data. We are just looking at different meta-analytically defined networks. And as you can see here, there are two things we need to appreciate. In that radar plot, Z transformed the significance for the classification. So the further out on the radar plot are, the better the classification patients versus controls. The red dashed line is the corrected significance level. So we do need to acknowledge that basically every network can detect both disorders above chunks. And that makes sense, right? Because they're both very systemic disorders. But on top of that, we also have a clear network disorder differentiation. There are some networks here in blue, like autobiographic memory, motor, semantic memory, that are much better in classifying Parkinson's than they are classifying schizophrenia. And conversely, there are other networks on the left side, emotion, empathy, cognitive control, that are much better in classifying schizophrenia than Parkinson's. So this opens the possibility that we have the same simple acquisition, but then we have different network models acting basically like probes into the data, looking at specific well-defined, a priori well-defined parts of the data that can then be used to extract different information also on the individual level. I'll skip that and just show you another example here on cognitive functioning in normal subjects. So can we predict executive function levels in normal controls? Here, two networks, the cognitive action network, cognitive control network, and the social affective network. Again, from the same resting state data, we extract two different functional connectomes, and then we predict on the individual level the spatial working memory by the Corsi block tapping task. And as you can see, if you use the cognitive network, you can actually predict individual cognitive performance quite well, whereas you cannot do this if you use the socio-affective network. That again underlines how important it is to extract the right information from a broad acquisition such as resting state. Last but not least, I want to outline sort of a rather generic pipeline of how this kind of biomarkers can be discovered, validated, and enriched. And I should have added this here. You can actually find this paper now on bioRxiv. Chen is the first author here, where we looked at 4 different psychopathology dimensions in schizophrenia, and then we tested a whole range of network models. And if you do that, you can basically get this result from a large fishing expedition. So you have a lot of different network models, 4 different behaviors, and as you can see here, there are quite a few that become significant. Now what we used is now a stepwise procedure. First of all, it was a multi-site data set, just 10-fold cross-validation. That gave us a solid amount of potentially associated or potentially predictive networks. And as the next step, we used a leave one side out validation. So whether within our larger sample, it could generalize to new site. That narrowed it down to 6 predictive associations. And then we used a completely new data set. I think MGH was actually involved, the BSNP data that we did not have access to before we actually ran the study. And then we tested the last 6 predictive models that survived both tenfold and leave one side out. And we actually found that out of this huge fishing net that we threw out, only 2 predictive models generalized in the tenfold, in the leave one side out, and onto the new independent data. And the top hit here was the association between the extended social effective default and the cognitive network. And the results are shown here. So these were the 6 network symptom associations that survived our internal discovery and validation, but only the 2 on the right one, they actually were also significant in both sides. So we made our life dedicatedly hard. So we independently tested the Hartford and the Baltimore side, and it survived significantly on both sides. And you can see here the scatter plot, actually even on subjects that were scanned at a new scanner, that were diagnosed in a different healthcare system, that came from a different population, we could actually predict individual symptom dimensions with a R value of at least 0.31. That's not something you base a clinical diagnosis on, but I think it shows the overarching idea of network discovery, replication, and validation. And last but not least, also what we show here is that you can do additional enrichment by looking at relationship to, for example, receptor transmitter systems. So we can show that those predictive models that survive also relate to both the serotonergic and the dopaminergic system. What's the overarching idea? The pitch I want to make? I think mapping the human brain, understanding brain organization, modeling inter-individual variability, and developing approaches towards personalized brain medicine. They are closely intertwined challenges, and we need to advance on all 3 levels to actually try and achieve the bigger goals of quantitative markers for brain health. And to do that, we need 3 components. On the technical side, it's large data sets with then the automated capacities for data management and processing. We need a priori knowledge on brain organization through atlases and functional data. And we obviously need the high throughput analytics and simulations environment that finally allows us to perform machine learning of variability and brain behavior relationships in both health and disease. Thanks a lot. Thanks a lot to you for listening, obviously. Thanks a lot to our funders, collaborators, and the wonderful people in my institutes. Happy to take any questions.